Hi, I'm Dr. Ayda Başkır Martin, and in this brief video, I will summarize the cell cycle for pre-nursing students. Cell cycle is a series of changes or events that a cell undergoes when they grow and develop from the time they form and the time they divide and have two daughter cells. Stages. Cell cycle has three main stages. Most of the time in pass through in cell cycle we call interphase, a preparation stage. Second is the mitosis, is a stage that cell DNA or cell nucleus divides. And third and the last stages is cytokinesis. We will call the cell cytoplasm divide. Now, as you see, we gave the like a knife cut stages: interphase, mitosis, cytokinesis. In reality, they are interconnected. Before one ends, another starts. So, but again, by order, you need to know the interphase, mitosis, and cytokinesis are the stages for the cell cycle. And you see in this graphic, and there's three checkpoints, and with the red color coded. And two of them goes in the interphase, the preparation stage. One of them is in the mitosis stage. And those checkpoints are important because it will decide the future or fate of the cell cycle or that cell. If there is something wrong with related with the cell or the growth of the development of the cell, and that checkpoint will either recess the development, recess the cell division, and the cell will stay in latent phase, or will decide to go further and go to divide. And it can also may decide if something really wrong related with the cell, and it may cause start the programmed cell death, which we call apoptosis. So apoptosis is programmed cell death, and during these checkpoints that can be decided as well. Interphase thought to be a resting time in past, but we know for certain that interphase is not a resting time. It's a very active period in cell cycle. It's the time the cell grows, it's the time cell maintains their normal function, and they replicate the genetic material, DNA. So DNA replication happens in the interphase, specifically in the S or synthesis phase, because interphase has three phases: synthesis phase, G1 and G2 phases. G1 or G2 phases either called growth 1, growth 2, or gap 1, gap 2 phases. So when we look at that, the cell replication happen during the S synthesis phase of the interphase. Cell synthesis of the organelles and membranes during the G1 or growth 1 or gap 1 phases. During the G2 phase or gap 2 phase, and all the biochemical material requires to the cell division time and produced in the G2 phase. So the interphase stages in G1, S, and G2. G1 organelles duplicated, S phase DNA replicated, and G2 phase the biochemicals produced to get ready for mitosis. Mitosis is the stage, cell cycle stage that produce two daughter cells from an original somatic cell. When we say somatic, that means it's not a sex cell, because it is different. It's included the meiosis, which is, we are talking only mitosis here. Mitosis, division of nucleus. As you will see here, there's some stages that we will talk: prophase, metaphase, anaphase, and telophase, and is the stages of the mitosis. And the cytokinesis is the division of the cytoplasm. It's shown in this picture as well. I think it's a very good picture to know those stages. As you see, it's a continuous, it's a cycle, it continues, it circles. Right? A cell grows, develops, divide. Again, the same cells, again, grow, develop, and divide. What are the phases of mitosis? We have four main phases that we will talk: prophase, metaphase, anaphase, telophase. You need to know them by order. So how to easily remember? I would suggest remember the word PMAT. You can form the PMAT. I color coded with the red color, and they are first letter of the words, right? The prophase P, metaphase M, anaphase A, and telophase T. And what happened in prophase? We said the first stage for mitosis. In this stage, chromatin condenses and form the chromosomes. So chromosomes formed and able to see it during the prophase. And nuclear envelope fragmented and nucleolus disappears. And when we look, centrioles migrate to the poles and they start to produce spindle fibers. So they have a specific, they look like a stars, each of them. We call them asterisks. So centrioles started producing spindle fibers and to get ready to the next stage, which is the metaphase. During metaphase, you will see the centrioles now in the two different poles across the cell, and spindle fiber is formed and chromosomes lined up along in the middle of the cell or equator of the cell. So M middle. It can remind you another easy remember what happens in metaphase. Chromosomes line up in the middle or midway. Anaphase, and the chromosomes goes away from each other, right? Sister chromatids, chromosomes separated, and they go to two different opposite poles. That is the anaphase, goes away toward the centrioles. And telophase is the last stage of the mitosis and or mitotic division, and chromosomes return to chromatin structure, and nuclear envelope started to forming, and nuclei become visible, right? And spindle fibers are disappeared, and cytokinesis starts after the telophase about to end. How our body control of the cell division. Frequency of cell division is strictly regulated and varies by cell type. For example, a skin cell, intestinal cell, and blood-forming cells divide often and continually. So if you damage your skin, and you will see your skin will divide and repair quickly, right? But if neurons damaged, and it will be difficult to repair, sometimes impossible. So neurons divide a specific number of times, then they cease to divide it. So you cannot repair it. That's why a person gets stroke, and if the neuron died, there is no another neuron comes and replace it. It's gone. It's gone. So there is different cells has the different type of regulations. When we look at chromosomes have their tips, right? And in this picture you will see the black color coded. Those end or tip of the chromosome called telomeres. And for every division, telomeres get shortened and shortened and shortened. There will be a point that there will be no shortening. Then they cannot divide, they cannot replace, and then life will end it. That's why the scientists and researchers is trying to improve and lengthen the life. They working on the telomere structure, because we call them mitotic clock, and determines our life expectancy, right? So because in every division telomeres will be shortened. And fluctuating levels of certain proteins in cell again control the cell cycle. Cell divide to provide a more favorable surface area to volume relationship. What that mean? If there is a smaller cell and instead of having one big cell, if there is two small cells, and you will have more surface area, more plasma membrane to do the processes. And what else affect the control of cell division? The hormones, right? Growth hormone, thyroid stimulating hormone, etc. And you will learn in the future. And growth factors are external controls for the cell division. Then when we look the how a normal cell divides and not become the tumor, right? And because we know that tumors are immortal cell, they divide uncontrollably, right? How the normal cell knows to divide a certain time and wait, but tumor or cancer cell doesn't know to stop. And because they have, normal cell has contact inhibition. Healthy cells stop dividing when they become crowded, but tumor cell does not. Tumors can result from a loss of control over the frequency of mitosis. When the control of mitosis, frequency of mitosis is lost, that means that cell becomes a tumor. There will be two types of tumor, either benign, that means remains in local area, it lost the contact inhibition, it divides, but only thing it does, stays there and presses the neighboring cell because it grows, enlarge, but cannot go other part, cannot spread the other part of the body. Malignant tumors or cancers, they are invasive. That means they destroy the neighboring cells when they get into the bloodstream. Via the bloodstream, they can go all over the body. So we call this metastasis, right? There's two major type of genes that may cause cancer. We call this either oncogenes, that abnormal forms of genes. They are not normal, abnormal forms of genes that control the cell cycle, but are overexpressed. They are overexpressed. So if there's a cancer causing agent, we call them oncogenes. Oncogenes, they cause cancer. Like UVA, UVB light is what? They are oncogenes. Helicobacter pylori, it's a microorganism. When you have in your stomach and long time, they may cause the stomach cancer, right? Stomach cancer. So we call this oncogenes. And tumor suppressor genes, and they are normally limit the mitosis. They suppress the tumor formation, but if somehow they are inactivated or removed, they cannot regulate the mitosis and the tumor forms. So cancer cells, for that reason, called immortal, since they do not stop dividing. Normal cells stop dividing after 40 to 60 divisions, but tumor cells does not. And tumor cells are so greedy. They produce their own blood vessels by releasing some angiogenetic factors, and they allow more blood cells. They produce more blood cells and more blood vessels because they need more blood to nourish themselves and grow exponentially. Before talking about the stem and progenitor cells, I wanted to give this picture to take your attention about the totipotent, pluripotent, and stem cells idea. As you see at the beginning, we had the fertilized egg and then the cell to form the embryo and placenta. We call them totipotent cells. That means those cells can specialize and become any cell type is possible. So fertilized egg or cells of the early embryo are totipotent. They can be any cell possible. And as you see later on, there is a pluripotent cells. They can be some certain type of cells. So they cannot be all the type of cells, but the certain cells that they can develop and become. They can be either blood cells, they can be either the neuron cells, or they can be muscle cells. So they specialize for the daughter cells. They have the limited number of cell types, not particularly one type, but limited when they are pluripotent. Now let's go see the definitions. So process of specialization of cell we call differentiation. So not all the cells in our body is the same because they have different function, different structures, although they have the similar organelles as we talk in the cell structure. The stem cell is the cells can divide to form two new stem cells called self-renewal. They can also divide to form a stem cell and a progenitor cell. Then they differentiate as any of many cell types. So that's how a surgeon can take from the bone marrow cell, stem cell, and they can manipulate in the lab, and they do the engineering, and they can produce a skin cell. So to use a burned patient to cover their skin. Progenitor cells partially specialized stem cells. They are daughter of the stem cell. They can divide to become any of restricted number of cells they call the committed cell. Differentiation is the maturation and specialization of the cells. A fertilized egg has the ability to produce any type of human cell, while partially differentiated cells can only produce some types of a single type of cell. Regenerative medicine is a branch of the medicine. It's a field of study that uses the body's ability to generate new cells to treat diseases and injuries, includes the stem cell technology. So when we talk about the donor stem cells, such as umbilical cord cells, they use the donor stem cells to treat the diseases and injuries. Stem cells from a patient, either from a natural site, such as a bone marrow, or reprogrammed differentiated cells grown in a cell culture and used in the regenerative medicine. Now, in my last slide, I would like to talk about the cell death. We call, if it's programmed cell death, it's a normal and it's protective terminology we call apoptosis. It's a normal part of development. It's a continuous stepwise process that removes, for example, removing webbing between the fetal fingers and toes, because at the beginning we have to float on the amniotic fluid. So between our fingers we have webs, but when we're born, we don't have web. So what happened? It happened because it apoptosis, and during the fetal development, and they disappeared. And again, if you have a sunburn, what happens? And your skin start peeling, right? Because it's protective. It's apoptosis. It damaged skin cells. It's programmed to get dead, and they peel off, so you will have underlying normal undamaged skin cells continues to protect your body. However, there is another word we call necrosis. This is the cell death from a damage, right? From a damage. This is not normal. This is a pathological cell death. So programmed cell death, normal, we call apoptosis. Cell death from a damage, we call necrosis. Okay, I hope you enjoyed, you learned. Have a good day.